Very good evening, Ambika. Okay, so I hope everyone join us. So let's continue. Very good evening, everyone. It is going to be our last session on the biomolecule chapter. Okay, so in the previous class, we discuss about the protein. There we discuss about what is protein, what is polypeptide, and all this thing. Okay, I'm sorry. So we already covered the protein part there, and then we discuss about essential, non-essential, every kind of that is the protein. Very good evening, Niharika. Okay, so let's continue further. In today's class, the topic for discussion is going to be the hormone, enzymes, and all this thing. Okay, so let's continue with the next topic. It is going to be the enzymes. Okay, so as you know from the chemical kinetics, enzyme is nothing. Enzyme actually the biocatalyst. Okay, that in biological chemical reaction, if we are using a kind of catalyst, so actually they are known as an enzyme there. So every particular reaction has a particular enzyme. Like if you want to convert the sucrose into the glucose and fructose there, so we are using invertase as an enzyme, right? So enzyme play a very much important role. They are just catalyst only, and hence since this is a catalyst, so they are behaving like a catalyst. Okay, in the which chapter surface chemistry part we already covered the enzyme chapter in very much detail. There we discuss about selectivity or like specificity there, optimum pH, optimum temperature, everything we already covered. Okay, so in today's class this is just a brief. What is enzyme? Okay, so detailed portion already covered in the surface chemistry. So what is enzyme? The reaction of the cellular metabolism are mediated by the remarkable biological catalyst and called enzymes. Almost all the enzyme are globular protein, if you remember that we discussed about two kind of protein in the previous class, fibrous protein and globular. So fibrous is insoluble in water, globular is soluble in water like the albumin. Enzyme are very specific. You know, in our body there is a n number of reactions going on. It's not like one enzyme catalyze the other reaction. You know the uh, specificity of the reaction there. Okay, that what is the meaning of specificity? That a particular enzyme only catalyze a particular reaction. Specificity, selectivity, every point. Okay, so maybe one enzyme catalyze one reaction, maybe that enzyme is not catalyze the second reaction. So it's specific in nature. And the ending name of the enzyme is always ase, like maltase, oxy reductase is there. So this is a like biology concept again, that how to represent enzyme every time in chemistry or like in biology as well. The enzyme end with this word, that is ase. Okay, reductase, maltase, invertase. Okay, ase be like this is just a representation. Okay, that's it. This is all for the enzyme. Next topic is the vitamin. So what does white mean? White mean is been, it's not like that they are very important for our body. Very good evening, sir. Okay, so like why I'm saying that not very important there. Okay, so enzyme be like a very small thing, you know, that in a food the vitamins like a salt. That okay, salt presence is not important in food, but the deficiency of the salt is really affected. So same concept of that vitamin, that organic compound that require in our diet in a very small amount. Okay, and performs some specific biological function to maintain the optimum growth and health in our body. You know, let's suppose if we are having a deficiency of vitamin A, so we are suffering from that is a night blindness. If there is a vitamin B, so we're suffering from beriberi. For C, there is a scurvy. Okay, for D, that is our bone start weaken there. Okay, so there's a every vitamin has a particular function in our body for a particular optimum growth and health. And most of the vitamin can't be synthesized in our body. So that's why they're requiring from outside. As in the previous class, we discussed about essential and non-essential part. I told you that if something is not synthesized in a body, that really important for our body, and somehow we have to maintain that. Then how to maintain that? Simply just by using a kind of particular diet. So how to take the vitamin? Just from a regular diet. Okay, so diet is very much important. So proper eating and healthy food. It's not like anything you have to eat, just healthy food. Okay, so what is the classification? This is very much important slide. The question asked from this topic: what is vitamin? Vitamin is classified into total two part. That is fat soluble vitamin and water soluble vitamin. First of all, which kind of vitamin? That is A, B, C, D, E, and then K. Okay, so like it's a continue pattern, but K is a different one. Okay, so water soluble vitamin R, that is a you can say these are soluble in water, fat, and oil, but soluble in water there. Okay, actually it's a misprint. It have to be insoluble, like vitamin B and vitamin C. And it's not like every form of the vitamin B. Okay, so just write some important point. That is only the B6 form. Okay, B6 form, B2 form, and B12 form is water soluble. Okay, for the vitamin B. If this is not like we are not able to remember, at least remember that is a group B, that is the vitamin B and vitamin C. So you can made any mnemonic like water soluble, B, C, okay, whatever you like. And then next is fat soluble vitamin. So fat soluble will be like A, D, E, and K. So you can just remember this or this. Like remember water soluble B and C, okay, and then the next become obviously the fat soluble. And biotin, that is a vitamin H, is neither soluble in water or in fat. Okay, so it's like a separate kind of vitamin. It is insoluble in both. Biotin. Okay, so if let's suppose you're having a very good knowledge about diet and all these things, gym in, so you know that what is that is a biotin. So it's a vitamin H. It's important. Three things: fat soluble, water soluble, and the last one that is a not soluble in anything. Okay, this question already asked in the previous year, that this H part and this two question already asked. This is all for the vitamin. Okay, this is not a biology class, so just a brief thing we are going to discuss. Next, the next one is the hormone there. Okay, you know that in our body that hormone is generally secreted from the master gland, that is the pituitary gland. Okay, every hormone, that every hormone regulation in our body. So adrenaline there, that is the insulin there, or there's just so many. Okay, oxytocin hormone. What is the function of the hormone in your body? Hormone, it's a kind of they regulate growth in your body. Okay, every particular hormone has a function. Estrogen, progesterone, testosterone. Every hormone has a very important role. If there is a little bit deficiency, okay, we're not feeling well, and either even our body is not functioning naturally. So what is hormone again? Like what is the actually no? This is chemistry. So what is hormone really? So hormone are the molecule that act like a messenger. Okay, let's suppose you are having anxiety, or let's suppose fear. So that our pituitary gland be like, okay, adrenal release, so that is we are able to maintain that soak at that point of time. So it's not like they every time present in our body. It's be like some processes going on, okay, and then signal transfer to the brain, and okay, that hormone release. So that's why the name is what? Messenger. Inside the cell, not from outside. They are produced from an endocrine gland in a body and poured in the bloodstream there. Okay, so actually that most of the hormone released in a brain. Okay, brain, and there's a gland, that is the largest gland, and then they further transfer to the other part of the body just by the bloodstream. Okay? It's not like they are generated at a particular point. No, most of the time at a particular point, and then transfer further. Okay, that's why the heart pumping is important, because blood circulation, you can say. Okay, so, okay. Next one, hormone have a several function in your body. They help to maintain the balance of biological activity. First thing, and on the basis of chemical nature, we have to classify the hormone into three parts. That is a steroid, okay, polypeptide, and amino acid. So basically, hormone is a kind of protein only. Okay, steroid. Okay, so you know that the steroid play a very much important role again in our diet. Okay, and polypeptide and amino acid, maybe you heard about this, like this athlete takes some steroid. Okay, so they are bad for our body, but still have a very important role in our body. Okay, so this is all for the hormone. That's it. Next is the nucleic acid. This is important topic. So what is nucleic acid? Nucleic acid is a kind of chain. Okay, nucleic acid is a kind of chain. It's like a polymer. Nucleic acid is a polymer, you can say. Okay, now what is nucleic acid? So everyone DNA. Okay, at least you are familiar with this word DNA. What is the meaning of DNA? Okay, deoxyribonucleic acid. But if I talk about like how DNA play a very much important role, it's a transfer of gene. Okay, the DNA play a very much important role to transfer the gene from one person to another person. Okay, if you want to check it like a particular offspring belong to a like a female or a male, we are just checking for DNA. So what is so special about the DNA, that that simply the thing is what, a heredity material. Okay, that a kind of thing that transfer during the process, it's like a heredity material, you can say. Okay, so what is nucleic acid? Nucleic acid of a living cell is responsible for transmitting inherited character. A process is known as heredity. Okay, so simple that when some character transfer from parents to the offspring, young one, so that becomes simply that as a heredity. Like from generation to generation, there's some trait that we are getting from our mother and father only, right? Some like biological social effect can be there, but like most of the time heredity. You know, we are having a particular habit that belong to our parents. That's it. Next, the thread-like structure inside a nucleus of a shell. Okay, so in a, I guess in a 10th class, or I don't know, in ninth class, you're observing these kind of diagram. Okay, so this is a kind of shell, and inside there's a nucleus. It's like that in our chemistry shell. Okay, we are having a nucleus, same there. So this is present in a nucleus. There's a thread like thing, and chromosome. So how the chromosome look like, this is how the chromosome look like. Okay, there is some kind of structure like this. Okay, chromosomes. So there's a total 23 pair of chromosome in our body. Okay, out of that, that is a two other sex chromosome. Okay, that X and Y. You know, if Y and Y, there is a female. If X and Y, there is a male. Okay, so you know better than me, because I guess you know that reproductive system in 10th class. You just deal. Okay, so next, the chromosome made up of a protein. So again, chromosome is nothing, again a protein. So wait, this nucleic acid is actually proteins, we can say. But not only protein, there is some important point. And another type of biomolecule called that is a nucleic acid. So all these thing combine, chromosomes, protein, and this become a particular class of nucleic acid. Now, what is nucleic acid? So it's further classified into two part. The the monomeric unit of the nucleic acid, it is a polymer, I told you just, right? So what is the monomeric unit here? That is nucleotide. There is a two word. One is this, that is tide, and one is nucleoside. So this tide and side make a huge difference. Okay, so what is the difference? First of all, uh, wait, let me just move the next slide. Okay, so what is nucleoside? Okay, so listen, a kind of nucleoside made up of three things, total two things. That is, one is a sugar, okay, plus nitrogenous base. So this is simple sorted, that is nucleoside. And if I'm using this word, that is nucleotide. So what is happening? It's the same. That is a sugar plus nitrogenous base plus phosphate ion. Okay, you know this is phosphate ion. So this is the extra point here. So I can say, I can say from here that I can say that is actually nucleotide is what? You can relate the thing. So nucleotide is actually what? This is a combination of nucleoside and plus phosphate. So be noted, for nucleic acid, the monomer is nucleotide, not the nucleoside. Okay, so it's really very much important part. So let's just back again what we are discussing. Okay, so on the slide, yes, here. So what is nucleotide now? That is a five carbon monosaccharide. If you don't know that sugar, we covered in the glucose topic. That I told you there's a five member ring have to be there, and there is a one more important point there, that this place is occupied by only hydrogen. Only hydrogen in the last, I guess. Let me just check this slide if I'm having. It's protein. Yes, look at this. Okay, remember in a glucose part we discussed, if you have to replace this OH with the H there, so it's become deoxy. It's a pentose sugar. So okay, it's a hexose, I know, but try to understand my point, that when you have to replace OH with the hydrogen, so it's become a kind of pentose sugar there. Okay, here. Look at this. First of all, so in this case, that is a five-member monosaccharide. Then a heterocyclic nitrogenous base. It is further classified into the purine and pyridine. Okay, so we will cover that part as well. Wait. So this is how the nucleic acid really look like. Okay, guess it's small. Okay, now it's visible. So look at this in this case that it's this is very much important. This belong to RNA and this belong to DNA. Okay, so maybe you're like, ma'am, structure is not important, then why you're wasting our time, right? Listen, my point. I know it's not important, but still, okay, this is a difference. Like, if let's suppose in a paper, okay, they are not asking about a structure, they are given a kind of structure, and they be like, okay, is this DNA or RNA? At least we have to know that what is the basic difference between RNA and DNA. So in DNA, that is only two hydrogen present at the second carbon, but here that second carbon has a OH. Okay, so when I was a child, I used to remember be like that is RNA used to drink. That's why that one have an extra OH group have to be there. Okay, so be noted, RNA extra OH group, DNA there is only hydrogen present. So what is DNA? Deoxyribonucleic acid. It is actually the sugar moiety. Okay, so what is this word moiety? Like structure. Okay, if you're not comfortable with this, leave that. Just remember that it's a sugar is beta D 2 deoxyribo ribose there, and this is beta D ribose only. In this case, what is happening that at beta position, this is alpha and this is beta position, so there's a 2 deoxyribose, and this case only beta ribose is there. Okay, this is the only point here. Next one is nucleic acid. So this part we just covered, right? This and this already covered, so it's a repeated thing. Let's go next. That it's consist of total three part, right? One is the sugar part we covered, that is either the deoxy or either the ribose. So there's a two kind of sugar. Now in the next point, that if we are discussing about here, that is the nitrogenous base. So it is further classified into two part. Wait, let me just write that nitrogenous base. I said nitrogenous base. It is further classified into two part, that is one is purine, okay, and one is pyrimidine. Okay, so this is purine and this is pyrimidine, two thing, purine and pyrimidine. Okay, next that it is further classified into that this consists of adenine, guanine, and if we're discussing about this, it consists of cytosine, uracil, thiamine. Okay, don't worry, we are having everything. Just try to understand. So, like pyridine is a 3, okay, purine is a 2 there. Now, in a DNA, okay, like DNA, RNA both are a 4-4 combination. So, DNA 4, and RNA still 4 there. But what is the basic difference between them? Let's try to understand difference. So, let's just back here that adenine, guanine, cytosine, thiamine, and adenine, guanine, cytosine, and uracil. See this or notice this thing that in DNA we are having adenine, and in RNA adenine. It's a common thing. Guanine, it's a common again. Cytosine is a common. How the DNA and RNA differ from one another just by this nitrogenous base. That in DNA you are having thiamine, and in case of RNA we are having extra, that is uracil. Make this point as a knot. It's really very much important part that DNA, thiamine, and RNA, it is going to be the uracil. Okay, so this is important, very much important. Okay, so let's go like how they really look like. Okay, so see this that adenine, guanine, so they're the part of purine. Okay, so purine part, these two, and this is the pyrimidine. Okay, so how to remember? Sometimes there is a problem, ma'am. How to remember? See, I used to remember be like in in the spelling of this pyrimidine, there is a M. M means mono. Okay, so all three are mono. One cycle, two, all are the monocyclic. But purine one, that is a two cycle have to be there. Okay, again, structure is not important. Just take a quick recap, okay? Just that if let's suppose if it is given in a paper, then how to deal. So pyridine, that is monocyclic, and the purine is a bicyclic, you can say. Okay, again, structure is not important, not important, and not important. Okay, so what is the point of to be noted here? That ACG, okay? I don't know the mnemonic in English, but I used to remember like that is for this purine, that is puri, aloo, and gajar. Okay, it's be like this is a dish. I don't know that how to remember in English, so it's be like P, A, and C there. And this side pyridine, cytosine, thiamine, and uracil. Okay, so this is how you can remember. Okay, then next point, like do you know how they are connected with one another? Okay, so just think about it. There's a kind of two classes. Okay, in one class there's a and G there. Okay, so A and G. Okay, both are bicyclic, and one class consists of C. Okay, and T and U. So how they're combined? A combined with C. Okay, AC and GT. If we are discussing about DNA, so AC and GT. Okay, I'm sorry. I just replaced AT and CG. Okay, so this is AT and this is CG. So this is they are combined with three triple bond, or you can say this is a double bond. And these bond are nothing. They're actually just a hydrogen bond only. So A combined with T, G combined with C. Okay, so do you know how the sequence in DNA? So the sequence be like, if let's suppose this is A, C, T, G, A, C. Okay, this is a some strand is going on. Okay, I will tell you how to work. Wait. Okay, just try to understand first of all. So this is a daughter DNA, let's say. Okay, so it is further combined with that A. Okay, so we know A combined with T. So the next sequence is T. So we just discuss C combined with G. So next sequence is G. Then T combined with again, that is A. G combined again with C. A combined with again T, and this is C, and the next is going to be the G there. So do you know if you are having one single strand of DNA, we are able to generate the second strand. Okay, and this is actually the scenario how the DNA transfer from a human being. So let's suppose this is your father. Okay, so every human being has a double stranded DNA. Okay, so let me tell you, that is this is how the double stranded DNA look like. Okay, this is a double stranded DNA. It's like a kind of ladder. Okay, so let's suppose this is A, this is T, double bond, this is C, and this is G, triple bond. Okay, so now this is how they are just connected with one another. Okay, so what is happening? First of all, let's suppose this is father. So one strand, let's say this is A and this is B. So this is my A and this is my B. Okay, then sequencing the another DNA and B sequencing the other DNA. So maybe in movies you are observing that if you go with the DNA fingerprinting, so the match is not fully 100%. Okay, every time that in most of the time in crime movies, it's going to be the 99.9%. Okay, still it's not 100%. Okay, that how many chances of a offspring to belong to their parent there. Okay, so this is actually how the thing really work in a DNA fingerprinting. Okay, so this is the point number one. And yes, from this topic the question asked in a paper, you are given with a sequence and there is some option is given to you, and question asked what is the correct sequence. Okay, yes, the question asked from the DNA topic. Okay, some sequence given to you, so I don't know how to remember. Just go for some mnemonic. Just remember that is ATCZ. ATCZ is the sequence we are going to follow. Okay, this is the only important point we are going to remember from the DNA part. Okay, other structure and all these things not very much important. Next, that what is nucleoside? I just told you what is nucleoside when phosphorus is not have to be there. So a unit formed by the attachment of a base at first position of the sugar, and one more thing, that is the base is connected at a first position. Okay, and phosphorus group connected at 5 and 3 position. Wait, we will cover that point as well just in a while. So in nucleoside, the sugar carbon are numbered as 1, 2 and 3, etc., in order to distinguish between from the bases there. So B noted first position is occupied by the base. Okay, if in an option you are given with 1 5, so it's wrong because one position always belong to the base. Okay, always belong to the base. Anyway, this is a typical nucleotide look like. So the first position occupied by a base, fifth position or third position, two position is occupied by a phosphate group. Okay, so in actually in DNA, how it is right that most of the time the direction is 3 to 5 or 5 to 3. So this is the only sequence pattern we are going to observe. 3 to 5 or 5 to 3. Like ma'am, what? No, we are not getting. So listen, that is this is a sugar. Okay, this is a sugar. First part combined with the base. Okay, so let's give the numbering. It is a 1, 2, 3, and 4. So it is combined with the phosphate group. Okay, so this is combined with the phosphate group like this. So this is a DNA. Let's suppose a single DNA is there. So there is a one more OH have to be there. So next phosphorus come, okay, and phosphorus simply combine here. So this is the sequence that only the phosphorus as a choice either combine at a third position or a fifth position. So you know that let's suppose what I want to convey. For example, if I write that there is a sequence 5 dash, okay, and there is a A, T, C, G, okay, and ta, okay, and this is 3 strand. This is my question, okay, I will give you, this is my question. Can you just tell me the resulting DNA for this? So what is the answer for this question? Be noted that if I'm having a 5, okay, be noted how to answer this question. If I'm having a 5, so this should be connected with the 3. So the next answer for this 3, A combined with T, T combined with A, G combined with C, C combined with G, and A T combined with A, and A combined with T, and this is 5. So this is the matching DNA for this pattern. Okay, so phosphorus group always combine at a fifth position or a third position. This is very much important. Okay, this kind of this is how we are solving the question from this topic. Okay, so I guess so much time. Now your turn. Is this point clear how to solve this kind of numerical in paper from the DNA chapter or the pattern? Okay, yes, prime you can say, Ambika. Okay, so actually what is it? It's a connection. Who's going to connect really? A phosphorus. Okay, so this is actually the important point here. So let's just back, and now let's move where we just left. So look at this. Fifth position already occupied. Now this is a kind of daughter DNA is going on. The next one is just fit at the third position only. So this is nucleotide. So now observe this, that how the typical structure look like. So this is a some DNA, okay? You know this is, let's say this is where it's connected, fifth position. Okay, and this is fifth position. Now they are having only the choice. This phosphorus only attack at the third position there, and this is how they are connecting. Okay, so this linkage, if you are observing the phosphorus, this linkage is known as a phosphodiester linkage. I'm repeating one more time, why the diester? Because you know ROR is a ester group. So this is my oxygen, this is my oxygen, two oxygen. So diester, and this is phospho group. So it's become phosphodiester linkage. Okay, so this is the fifth end of the chain, and this is a three end, and it will continue like that is a 3 5 5 3 again. See, this is 5 3, and then up to so on. So sequence be like 5 3 5 3 5 3 or 3 5 3 5 3 5. Okay, so it's not one, be noted, because one position is always occupied by the nitrogenous base. It is very much important thing. Next thing, okay, so now what is the DNA structure? So there is a so many n number of scientists come and give different structure, but the only famous structure of the DNA given by the Watson and Crick. Okay, so it is a double stranded helix structure. Okay, and it is given by that is a James Watson and that is a Francis Crick, famous on the name of Watson and Crick model for DNA. Okay, so in this case what is happening, two nucleic acid chain are wound or like simply combined with hydrogen bonding. Okay, these nitrogen genus base, nothing has a free OH group, they are forming a hydrogen bond only. Okay, so let's see actually how a typical DNA structure look like. This is that double stranded uh, structure of DNA there. Okay, so this is a helix of the phosphorus. Okay, that is, this is a phosphate group, this is a phosphate group, and in between that we are just fitting, let's suppose this is A, this is T. Okay, so this in between nitrogenous base are just connecting. Okay, this is a helix kind of thing. And what is the theory? Two standard complementary. What is the meaning of complementary? If let's suppose my one chain is running from 3 to 5, so the next is 5 to 3, like opposite. Okay, we just discuss A become T, T become C. Sorry, T become A and C become G. Okay, so just complementary to one another. So adenine form hydrogen bond with thymine. Between adenine and thymine, there is a two hydrogen bond, and between cytosine and guanine, there is a three hydrogen bond. Okay, this is all for the DNA. This is important topic from the board exam point of view. That is the structure of the DNA. Okay, yes, most of the time this question asked in board exam, explain the Watson and Crick model of DNA. So how to write? That is the double stranded helix structure. Okay, the phosphorus is forming the helix. In between the sugar is there, and how they are connecting. These things you are going to write in a paper. Okay, so this is all for the DNA. Now let's see the next one, that is RNA. Okay, so how about the RNA there? RNA, it's not like double stranded. RNA is a single stranded. That's why that RNA is not a heredity material. Okay, yes, in some prokaryotic, that RNA can be the material because they are not that much developed, single cell organism. Okay, single cell. Okay, no sorry, that no nucleus is there. Pseudopody amoeba, I guess you know, right? From 8 to again 9th or 10th, I don't know which class. So the single stranded. Okay, it's a single stranded there, single strand only. It's not like DNA complement. Okay, so a single zigzag pattern is going on. So like in the structure of RNA, helix are present, which is only single stranded and sometime folded back. Okay, let's suppose the single strand is going on and it's be like, okay, I'm very lengthy, let's again fold back. So only one thing is just keep rotating there. Okay, and if double strand helix, so that a single chain itself tangled with one another. So that's it, this is all for the DNA RNA. Now important function of the nucleic acid to protein synthesis. Okay, okay, let me just give you this point as well. Okay, this is a very famous process if you want to convert a DNA into RNA. Okay, DNA, actually RNA is the, that you can say cellular level. It's okay, DNA is our heredity material, but in our body actually RNA is going to work. Okay, in our body the last form is what, amino acid. Okay, DNA is not that much stable, RNA is not that much stable. Actually, like if you have to compare between DNA and RNA, so DNA is much more stable over RNA because RNA is a single stranded. So conversion of DNA to RNA is known as a transcription. Okay, transcription. So you can remember, be like transcript. We are just simply changing, okay, completely changing DNA into RNA. And next, now RNA convert into the amino acid. Okay, so this process is known as a translation. This process is known as a translation. So translation, when you have to translate something, RNA is a protein. You are translating the protein, the smallest unit of the protein, amino acid. We discuss about alpha amino acid. So translation, RNA to alpha amino acid, DNA to RNA, it's a transcription. Just remember the name of the process, it's enough. Okay. And if we're discussing about the type of RNA, so there's a total three type of RNA. rRNA, okay, that is ribosome RNA, and that is mRNA, messenger RNA, okay, and then that is a tRNA, that is a transfer RNA. So they are having a particular role in our body. We are not going to discuss in very much detail because this is not a genetics class. Yes, Ambika. Okay, so this is not a genetics class. So leave that. Just remember this point only, DNA to RNA to transcription to translation. Okay, so let's back toward the RNA structure. I know it's be like so much information. So this is RNA. Okay, that's it. Let's back again here. So protein synthesis, an important function of nucleic acid to protein synthesis in a shell. The protein are synthesized by the various RNA molecule in a shell, but messes for a synthesis of a DNA. Actually, I just told you mRNA. What is mRNA? Messenger RNA. So if let's suppose this is DNA, this is RNA. So this RNA be like, okay, let's suppose this is a shell number A, this is shell number B. So this is transferred to the cell A. Okay, B required this. So A transferring the message there. So messenger, okay, in between be like, because DNA be like, I am the boss there because I am stable. RNA you're not stable. So you can be a worker. Okay, so RNA just working in between the shell, transferring the message. Okay, that's it. So RNA molecule in a shell, but masses for the synthesis of particular protein present in a DNA. Be noted. RNA is a messenger, but the masses, the thing who's going to handle the DNA. So DNA is like a boss who have a information. RNA is just a messenger. Okay, and one of the best application that is the that DNA fingerprinting. Okay, maybe you know that is a blotting techniques. Okay, if you are having okay, you are watching the crime series or the shows, so you know that let's suppose there's a person, okay, there's some crime is going on, and then we're collecting some sample like hair samples, saliva, and all these things. Okay, and then we are using a kind of thing, and then okay, that is known as a DNA blotting. This is not the topic discussion right now. So now the question be like, what is DNA fingerprinting? What is DNA fingerprinting? Okay, so like how it is possible, let's suppose this is a sample and it is belong to a particular only a one person, not for every person, even twin have a different DNA. Okay, detail already. So you are a biology student, Ambika, I guess, right? So I guess you know that. So anyway, what we're discussing, yes, my question to you, what is so special about the DNA? You know, there was a statement, if DNA keep increasing, there's a point came into the picture that when the complete earth is surrounded by the DNA only. Are you getting this statement? It's not a statement, it's actually a very bizarre statement. Just think about it. Okay, let's suppose there is a one person, it give 3 to 5 strand, then a different person, person number 2, then 5 to 3. So every person has a, you know, so many DNA and so many pattern in the world. So listen my point, as I already told you. Let's suppose this is a father. Let's suppose this is a mother. Half stand come and combine. So they belong to a single person only. So single person means DNA fingerprint is a identity of a person. It can't be copied. Even the twins have a different DNA. Be noted this. Anyway, what is it? What is DNA fingerprinting? Every individual have a unique DNA fingerprinting which have been used for the identification purpose for a long time. We know, right? The information regarding the sequence of a base on a DNA of a person is called DNA fingerprinting. That's it. It's important. That much statement important for the chemistry. Okay, we are not going to in very much detail that we are using some kind of enzyme for breaking. Okay, no, leave that. Thank God we're not biology student. So let's back. Now, task for the protein. Okay, these tests are very much important. Dihydrin burate test, and that is a xanthoprotic test, seric ammonium test, and carbalamine test. If you are not able to remember the test, no, not an issue. But remember the test, that's ninhydrin test for which protein, polypeptide, amino acid. Burate test is for amino acid. Like at least remember the substrate only. Okay, it's not like that you have to remember reagent structure. No, it's not important. Just remember the substrate part only. So let's go with the first test, that is ninhydrin test. So what is ninhydrin? That it is famous for alpha amino acids. So the point to be noted, ninhydrin test for amino acids. Alpha we can leave, but amino acid means ninhydrin. Okay, so this is alpha amino acid. So if you are observing like you know, with respect, this is alpha. So alpha amino means like alpha amino acid both are present. We discussed already this point. So this is the structure we are using now. This complex is little bit important. Okay, you be like, ma'am, you told us structure is not important, but it's important. Okay, so how to remember? It's not difficult. Okay, it's not difficult. Let me just form actually how it's working. Okay, then it is easy for you. Okay, so you know this is a six-member ring. Okay, it's a five-member double bond and carbon NOH in this. Okay, this is a normal structure. Now, do you know actually what is happening? Oh, I'm sorry, I missed one CO. Okay, do you know what is happening? These two OH is just replaced. Okay, these two OH is just replaced by a nitrogen. One point, and then same ring you are drawing on the next side. Okay, O and double bond O. Okay, this is a benzene there. Okay, so in this case, you are observing like if there is a double bond, it's like ketonal form. There is a negative charge. Okay, it's be like reverse. And if let's suppose if I'm using a double bond here, so this is my O negative. So this is. Just connect these two things. One is nitrogen, one is carbon, and this is how it's going to work. Let me just back again toward the structure. Okay, where was the slide? Yes, here. So this is actually how the structure formed. Okay, so this is a like purple color, and if purple color is there, again it's a biology test. Ninhydrin is a very famous test of a biology lab. Okay, it's a purple color pigment we are end up. Okay, again, if not able to remember anything, at least remember ninhydrin is for amino acid. Next test, burette test. This is important for polypeptides. Peptide bond, you can say. If you want to check it, like how many peptide linkages have to be there, so just go for that. Okay, this is copper positive. We are using copper is converted, forming a kind of complex there. This is a pale purple complex, ma'am. No need to remember the structure. No, it's not important. Remember that ninhydrin for amino acid detection, so burette test for the detection of peptides. If let's suppose in our option you are not given with peptide, so if you are given with protein only, so then go for the protein. If peptide is not given, if it is given already, okay, so then you have to select only the peptide. Next, that is a xanthoprotic test. Again, it is important. It's already asked in a previous year if you are solving the PYQ properly. Tyrosine. Tyrosine give xanthoprotic test. What is tyrosine? In the previous class, if you remember, I told you that it is a kind of aromatic amino acid. This is a C and this is H and this is NH2 group, and then this is a CH2 group and it is a phenyl group and it is connected with the OH group. I told you the structure in the previous class if you remember. So anyway, tyrosine, that mnemonic be like all trees are pine and timber are trees. Okay, so this was the mnemonic. So that tyrosine and a yellow color complex. Complex is important? No. Okay, again, test is not that much important. The name does. Tyrosine give xanthroprotic test positive. Next point, that is seric ammonium nitrate test. This is famous for the serine. Okay, serine. Serine is a alcoholic amino acid. Recall that is alcohol is a severe threat. That is threonine and serine are the two alcoholic that what amino acid. Okay, so in this case we are having cerium using cerium ammonium nitrate. This thing we already used in a previous chapter as well. And red color better if you are able to remember. If you're not able, leave that. At least remember serine gave the seric ammonium nitrate test positive. Okay, this is the key point. Next, that is a carbalamine test. This is not new for us. We know primary amine. Remember, primary amine give carbalamine test positive, and we are end up with the what? That is the isocyanide. It's a very that is obnoxious smell. Confirm the presence of primary amino acid there. Okay, primary amino acid. So this is KCl and water have to be there. Carbylamine test. This is all for the that which protein part. So let me quickly brief this. First of all, ninhydrin amino acid. Okay, alpha amino acid if you are able to remember. Next one is what? That we are having seric ammonium for serine. Like serine, it's a rhyme that seric ammonium serine. Okay, and then that burette test that for polypeptide, and this is the carbalamine. This is for primary amine. Okay, all are the tests for that nitrogen or you can say proteins. Now let's see the last topic, that is the test for carbohydrate. Okay, if you are having a very good knowledge of the lab, at least everyone you know at least Molis test. Okay, I'm not saying that you know Barford test or Sullivanov test, but at least Molis test you know. Okay, anyway, let's discuss one by one. What is Molis test? Okay, ma'am, it's difficult to remember. This is nothing glucose. Remember this structure I told you there, that when both all OH are equatorial, so it is which kind of alcohol, glucose? This is a beta glucose. Again, alpha and beta is not the topic of discussion right now. Okay, so we are having a glucose and we are using, okay, reagent you have to remember. This is important test. Molis test you have to remember. Alpha nephthol plus that is the concentrated H2SO4 and glucose, and we are getting the furfural. Okay, just remember the name. Furfural. Okay, and if you are able to remember the structure as well, so it's good for you. Okay, how it is formed, mechanism, no, it's not important. Moles test, you're having a glucose, alpha naphthol, concentrated H2SO4, we are getting furfural. It is a white color compound, violet color, I'm sorry, violet or purple color. Moles test. Next is the, that is a bar for test. Again, in this case, we are using copper. It give the positive test for the aldehyde presence in a glucose. Okay, so it is a kind of, if you remember the copper test, the brick red color. Okay, in Tolan test, we discussed this test, copper test there as well. Okay, so it is a repeated part. Only this is going to oxidize. If you're observing, this is going to oxidize. Copper, it is going to reduces there. Okay, and reddish brick red color we are getting for the copper. Last, very much important out of all the three tests, Sullivanov test, already asked I guess 3 to 4 times in a previous years. Okay, so what is this about? What all about the Sullivanov test? Okay, so the most commonly asked question from the Sullivanov test. Do you know this test given positive by both kind of glucose, ketosis like fructose or aldoses like glucose, but this test given fast by the ketose. If you want to differentiate between ketose and aldose, okay, I'm repeating again, if you want to differentiate between ketose and aldose, so ketose give this test fastly. Okay, so this is how you can distinguish them as well. So it's a fructose. Actually, what is happening, we are using concentrated HCl. What is actually, let's actually just for this reaction only, how the mechanism is there. You know, you're having carbon, this is further carbon, OH and hydrogen, this is one more carbon, OH and hydrogen. Okay, we're using HCl. So what is happening, that hydrolysis, this is releasing double bond. So it's be like that water molecule start releasing there. It is a kind of dehydration process. Okay, so every time if you're observing that all the carbon, okay, this is combined fructose structure we already covered, right? And then there is a carbon double bond, there's a carbon double bond, and this is ready. And actually, what is it? We just discussed this, that, okay, remember? Okay, we just covered. Isn't it that previous slide for fuel? Yes, the same thing. Okay, and if you are not able to remember that cycle, where was the cycle? Okay, this. Okay, just remember that. Okay, it is easy to remember even for fuel. And then we're using resorcinol. And again, reagent is very much important. Two reagent. First of all, we are using HCl and then resorcinol and red color. Okay, out of all three tests, or even out of all the that total four and then seven tests, this is very much important test, Sally-Venoff test. Okay, important. Resorcinol and HCl. We are using red color complex. We are getting ketose. Give this test fastly as compared to the aldose. Okay. So that's it. This is all for the test. This is all for the theory. This is all for the biomolecule theory. Now you have to form some chart or some notes and then revise them properly. Okay, biomolecule, it's be like if you're once you have to revise, and if let's suppose they fit in your memory, it is going to be the lifetime. Okay, important. Anyway, now let's solve some questions. I hope after so much discussion, now we're ready with the questions. Okay, please, it's a request, try to answer the question. If you don't know, at least try, okay? So let's start with the question number one. This is a question number one. Question asks, the peptide that give positive steric ammonium nitrate and carbylamine test both are positive. Both steric that steric ammonium nitrate and carbylamine test positive. Yes, please. Answer this question. So there's a key point, steric ammonium nitrate, right? So yeah, it's easy actually. Okay. Okay, so I hope this point is now clear for you. So we just discussed serine, okay? So just observe lysine, glycine, aspartic acid, ASP, lysine, and again aspartic acid, and that is a glutamic acid. See this, that is only this one has a serine. And how about the carbylamine? Primary amine have to be there. You know the structure of the lysine. Remember that glacier at Alaska. So that if we are trying to draw the structure as well, so how the structure look like, that this side we are having NH2 and wait, and then this is a COH and this is hydrogen and glacier at Alaska. Okay, so after an aniline, lie, okay, so it's a carbon, one more CH3, and this is CH2. So this is the structure of the lysine. Okay, important primary amine present. It give the test positive. Yes, it is going to give the test positive. Okay, structure not important. Again, not important. Okay, done. Option B is the correct here. Next question, please. Question asked, in both DNA and RNA, heterocyclic base and phosphorus linkase present at what? This is important question. Question asked about DNA and RNA. Okay, so if you don't know about the RNA, at least go with the DNA. Just by using a DNA topic only, we are able to remove at least two options. Okay, actually, my mistake. DNA, RNA part I am not highlighting there. So you said C1 and C2 respectively. Question asked about a heterocyclic base and a phosphoester linkase. Okay, so it means two things we have to identify. One is a base, one is a phosphorus linkase. Um, okay, very good. She's the correct answer here, right? Yes, this is my last session. Okay. So, heterocyclic base, first position we discuss, right? Phosphoester linkase, fifth or three. Okay. So here three is not mentioned, at least five were given. So option C, go with option C there. Okay. So that's it. Last question, important question. Let's see how many of you are able to solve this question. Okay. It is a main question. Okay. I guess 2019. Hmm. Yes, please. You have to solve this question from the topic of the previous class. Isoelectric point. I told you that pH increasing, decreasing, how the sign is going to be changes there. So question ask about, you are given with 3 pH, pH 1 that is 2, pH 2 that is 6, pH 11 that is okay 11 there. G1 is the charge when pH is 2, G2 is the charge when pH is 6, G3 is the charge when pH is 11 there. Okay, so first of all, out of pH 2, pH 6 and pH 11, we have to identify that which one is the isoelectric point. Then we are going to proceed, right? Then and then we are able to proceed further. So now, if let's suppose you are given with 3 pH like 2, 6 and 11, so the mid one like 2 acidic, 11 basic and the 6 is the isoelectric point. Okay, so the point number one we are selecting from here, that my pH 6 is the isoelectric point here. Okay, so out of from this point that I am able to interpret, that is isoelectric point is what, that is 6 point. Easy. Okay, now just try to observe the charges. Recall if pH is less, pH is 2. So what is happening there? That is a concentration of H positive and is maximum. And if H positive is maximum there, then what is really happening? Think about it. Okay, like positive charge, negative charge, cathode, anode. Recall the previous class. Okay, take your time. Okay, 1 minute and just try to answer the question. If you're not getting, don't worry, I will explain. Please solve this. It's a request. 8.5, okay. Question asked about the charge, okay? It's not pH. Ambika question not ask about the isoelectric point here. Question ask about the charges on a molecule. Okay, let me give you the brief. Actually, what is the hint? Okay, listen my point. That for example, if this is some kind of acid, this is hydrogen, this is some R, and this is CO and H, and this is NH2. So we just discussed that pH is equal to 6. This is my isoelectric point. Okay, at that point, Jupiter ion have to be there. What is the meaning of Jupiter ion? That is, this is N positive and this is CO negative. Okay, it means at that point of time, the charge is equal to 0 on the molecule. There is a no charge. If my pH is equal to 2, it means less, okay, or you can say very less pH here. So I can say at that point of time the H positive concentration is maximum. Okay, wait, I am changing the position. Okay, now listen. So what is happening in this case? Okay, so what is happening in this case? The pH is 2 and concentration of H positive and increasing there. So if concentration of H positive and increasing, so this be like I am negatively charged. Okay, if H positive present, so I have no choice. Okay, I have to take the OH. Sorry, I have to take the H positive. So it means at the low pH, this is neutralized. So we can say at low pH, only positive charge is active. Only positive charge is active. Similarly, if the pH is equal to 11, it means greater pH, or we can say concentration of H positive is very less. So now this time opposite, the ammonium ion be like, I'm having 3 H positive ion. Why not I give my one hydrogen so that we can balance the H positive deficiency into the solution? And just because of that, what is happening? That it become N2 there. It's negatively charged. Okay, is this point clear for everyone? Actually, what is going to happen? That at low pH, who's the winner here? That at low pH, positive charge really present, and at high pH, negative charge present. At isoelectric point, there's a no charge present. Okay, now let me just back toward the question again and now try to answer the question. This is the question. So in this case, observe that, okay, let me tell you one thing, that if my pH is 6, so my Z2 is definitely going to be the 0 there. Okay, this is a modulus. It means negative, positive, whatever be the charge, we are only going to assume the magnitude part here. Okay, so G2 is the 0. Similarly, you have to identify how about the charge for the pH 6 and how about the charge for the, sorry, how about the charge for the pH 2 and for the 11. Okay, I hope that you are getting my point, and now please try to answer. Still, if you are not able to do, I will explain, okay? But at least try, right? So, what is your point of view? For, okay, Aditya. No, 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 no. Okay, C2O means. Okay, Aditya, come on. Listen, that if you are having R CO2H, what is the meaning of this? This is nothing. CO and OH is there, right? Two oxygen. So it's a COH group, acidic group only. Okay, it's not peroxy case. Peroxy case means that when, let's suppose, this is R, CO, and then O, and then O, and then H. So you can say R, CO3H. So in this case, the peroxylin case have to be present. Okay. Okay, so you're partially right, Aditya. Let me tell you why, or how everyone, that how to solve this question properly. Okay, so listen my point. Let's talk about that when pH is equal to 2 there. So pH is less. Okay, so when pH is 2, pH is low, H positive concentration is high. Okay, So who's the winner? Think about it. H positive more, CH neutralize. So positive charge have to be there. So at pH is equal to 2, where the positive charge, this is free NH3. Okay, first, this is free NH3, 2. So how much charge? 2 positive. So we can say that is my G1 is equal to 2 there. Okay, modulus, so only magnitude we have to consider only 2. Similarly, that for the pH 6. Okay. For the pH 6, what is happening? There's a no charge in a molecule because Jupiter ion have to be there. So it's a 0 there. No charge. Now, if my pH is 11, so what is happening? pH is high. So then H positive concentration is low. So remember, negative charge have to be there. So what is happening at a high pH? Look at this. COH negatively charged. COH negatively charged. So 2 negative, ma'am. Answer is 4. But no, this is also the proton. This is also the acidic proton. Yes, actually we are habitual only about the COH and NH2, but how about this? This is also acidic in nature, so it's also give the proton. Okay, this is the only key point in this question. Otherwise the question is easy. So that's why my G3 is actually what? It's a 3. Okay, so what is the total charge? If we have to add the total charge here, that is Z1 plus Z2 plus Z3. So it's going to be the 2 plus 0 plus 3. So the correct answer for this question is going to be the 5. Okay. So this question clear for you? And yes, any problem if still you're having, so let me know. Okay. This is all for the biochemistry. This is all for the syllabus. Now at my end, syllabus is finished. And that's it. If you want to understand some point or something, just connect with me. And okay, that's it. Yes, please. Any doubt, any problem? No, not okay. Thank you so much then everyone for joining the class. Thank you so much for your time. And again, keep preparing. Okay, you will do. And all the best. Okay, thank you so much. No, Ambika, because syllabus is already finished. Okay, that's why. Because the strength like 6 to 7 students join in a live class. So like according to me, it's just a time waste for me. Because okay, the strength be like 30 something, so I can go. But no, right? Thank you so much then. Okay, my promise was only to finish the syllabus, right? So thank you so much. Thanks for joining. Thank you. Thank you so much. Have a nice day.